Muscle tissue. So the function of the muscles, we already know the basic functions of the muscle, which is contraction, of course. There are many different terms that we're going to see inside muscle. Of course, we discussed in the beginning, in the first practical, that some specific cells have specific terminologies for their intracellular function organelles. Let's see what actually we have in muscle. We have the term sarco. Sarco means in ancient Greek muscle. So sarcolema is the term that we use to describe the cellular membrane of the muscles. If the muscle, that can be either smooth muscle, cardiac muscle, or skeletal muscle. We'll discuss these muscles, the different types, specifically one by one. Then we meet the term Sarcoplasmic Reticulum. This is the term that we use to describe the smooth endoplasmic reticulum of the muscles. These terms actually have these names because they have a specific and different function than they usually and typically do. And the sarcoplasm is, of course, the cytoplasm of the muscles. Let's see the different types of muscle. The skeletal muscle, also called striated, the cardiac muscle, and the smooth muscle. So first we're going to talk about skeletal in detail, and then we're going to progress to the other two types. So the reason why it's also called striated muscle is because we can actually see striations in hematoxylinosin staining. It's actually a very, very typical and very indicative procedure and indicative picture that we can see. They're very rare, very rarely we're going to see these typical striations in the slides. So let's talk about how these muscles came to be. These muscles, in fact, these cells have the skeletal muscles actually have many nuclei. This is typical of cells. Again, we discussed this before. Typical of cells that actually fuse together. For example, on the osteoclasts, we saw how the monocytes fuse together to form the osteoclast. And of course, these cells have 5 to 50 nuclei. In this case, we have the myoblasts. Again, mesenchymal stem cells that eventually will fuse together to form these myotubes, and eventually these myotubes will become the muscle fibers. Now, some cells, some myoblasts differentiate into satellite cells. These satellite cells are nothing more than just progenitor cells and cells that can potentially differentiate to form skeletal muscle. Generally, tissues that have progenitor cells in their periphery or close to the alive and the functional tissue can present some sort of repair, some sort of regeneration. But again, satellite cells are not very active in the adult human body. So destroying, scarring, or somehow damaging the skeletal muscle will maybe provide some sort of, let's say, will regain some part of function, but not necessarily as it was in the beginning. So satellite cells are not highly functional in the adult human body, but sometimes they can actually restore the function. Again, not fully. So let's talk about how they are actually how the muscles, generally the muscles, are organized. Then we'll discuss how the muscle fiber is organized, and then we'll discuss how these structures that we'll see will actually come together to produce the function of contraction. So this is a picture that actually helps out a lot. We'll start by the muscle fiber in the beginning, the smallest unit, and then proceed to the bigger ones. So first we have the muscle fiber. The muscle fiber is nothing more than just this fused myoblast, very elongated, long structures that contain many nuclei. And of course here, this tiny structure right here, this yellowish structure, is the satellite cell. So the muscle fiber is encircled by the endomysium. The endomysium is specifically a connective tissue layer filled with reticular fibers and fibroblasts. So many different muscle fibers together, along with the endomysium, will be organized together to form the fascicle. So the fascicle is nothing more than just many muscle fibers aggregated into one specific location. This fascicle will be surrounded by the perimysium. The perimysium is just thin connective tissue, loose connective tissue specifically. Now, many fascicles together will again aggregate together, will come close together to form the muscle as it is. So this structure, the muscle itself, is surrounded and covered by the epimysium. This is dense connective tissue. Now, this connective tissue will be continuous with a tendon. The tendon, again, we saw it in the connective tissue lecture. The tendon is nothing more than dense, irregular connective tissue. So this connective tissue of the tendon will be continuous and one and the same with the epimysium. This is how actually our body The tendons can actually promote movement from the muscle to the bone and vice versa. So, very important to notice is that the muscle generally is highly vascularized, containing many nerves, vessels, and lymphatic vessels. Why is it important? Because, of course, the muscle fibers, the skeletal muscle requires high amounts of ATP, and of course, there is no ATP without oxygen. So high perfusion of this structure allow the muscle to produce a high number of ATP. So we have, again, these capillaries and nerves will be found specifically in the perimysium, around the perimysium, and tinier and smaller vessels, let's say capillaries, will be found surrounding the endomysium. So let's discuss what actually makes up a muscle fiber. There are very specific functions, specific structures that we'll see. Right now, first we'll discuss only the structures with just short mentioning of the functions. Then we're going to put all these pieces of puzzle together to see the general picture and the general function, which is that of contraction. So first up, we had this division when it comes to, which is done by visual, this is a visual criterion. We have these zones and discs. These zones and discs are nothing more than just a visual classification of what we see inside the muscle fiber. They do not necessarily affect structures and functions. This is not a functional classification. There is no specific function of the A band or the Z band or the I band. This is just something that we see visually and which simply name these locations. The contents of these locations, the content of the A band can have some functions, but not necessarily the A band. We cannot say that A band has this function or I band has this function. Let's talk about them first, and then we'll discuss and see why we cannot have this classification, why this classification is not functional, it's only visual classification. So we have these terms: bands, lines, discs. Band is just a specific area of the sarcomere. One second. Band is a specific area of a sarcomere. So the A band the name is called from anisotropic band because we find two different components inside. Three actually, but typically the most important of the two that we'll see. So the A band, the anisotropic band, contains both actin filaments and myosin filaments, thick and thin filaments. We already discussed filaments in the beginning of the cytoskeleton. Of course, it's very important to understand that the muscles are filled with actin filaments and myosin filaments. The actin filaments are what we call the thin filaments, and the myosin filaments are what we call the thick filaments. So after there's a picture lower, we're going to see it's much clearer what these bands contain. But let's talk about the terms a bit. Anisotropic band is because we have different, more dense locations, more dense content visually when we see this slide, when we see this portion of the cell. Then we have the M line. The M line simply connects the two different points of the A band. They connect the thick filaments. Then we have the I band. This is for the term isotropic band. This is because it's more not as dense looking. It's more loosened in the macroscope. And the Z disc is the connecting point between the two different sarcomeres. What is a sarcomere? Sarcomere is the beginning of one Z disc to the ending of the Z disc. Meaning what? The Z disc is it's from German, meaning between, and this is the Z disc, and the Z disc will form the sarcomere. We'll discuss why we have this distinction. What is specifically the sarcomere? So within this sarcomere, we're going to find all the different bands. We're going to find the half of the I band, of course, because this is where it actually starts. This is where it actually starts. The sarcomere containing half of the I band here, A band, half of the I band. Now again, the M line is the connecting point between the thick filaments, and the H zone is just one portion of the A band that is again loosened. Again, do not think that this is a functional classification. This is only visual. This is exactly why you have this classification, because in this microscope specifically, we can see this specific lines, the Z discs, clearly seen. This is part of the I band. This is the full I band, of course. So I band, Z disc, A band, and M line. So what are these made of? What would we find inside these structures? In the A band, in the anisotropic band, we find this mixture of the thin filament. This is the thin filament. This is the titin. We'll discuss what is titin. Titin specifically is actually the largest protein in the human body, and in this location, the densest, actually the one with the most daltons, the heaviest and the biggest protein in the human body is titin. Interestingly, the name titin comes from the word titan, which actually means very, very huge. So we have this titin, and this is the thin filament, the actin filament, and this is the thick filament. Because we see this overlap of the thin filament with the thick filament, this actually forms this dense area right here. Why it's actually called anisotropic band, because there is a dense appearance in the macroscope. In the eye band, the isotropic band, we only see the thin filaments along with the titin. So of course it's way less dense. This is actually why we have this classification, this visual classification, because we can see clear pictures of dense and lucent areas in this microscope. Now, let's focus a bit more in the structure of the actin filaments and the myosin filaments. Again, the thin and the thick. Let's talk first about the easier part, which is the thick. The thick filament, again, this is the myosin, is nothing more than just it's a cytoskeletal part that actually is responsible for the attaching. This actually attaches on the thin filament. These heads, these myosin heads, contain ATP binding sites. ATP, of course, we know this is the source of energy. So these heads actually contain the sites where the thick filament will bind to the thin filament. Why? We'll see why. But at this point, just know this, that the thick is actually has higher molecular weight, it's thicker, and of course contains it's a full portion of myosin heads. Myosin heads again contain the two different portions that we really care about, the ATP binding site and the actin binding site. Now, let's focus on the thin filament. This is actually the most interesting part to me, according to my opinion, of the muscle, the thin filament. Why? Because here we find three different units that play the biggest role in contraction. Of course, we have the F-actin. We discussed what's F-actin. F-actin comes from the word filamentous actin, which is practically a polymer of G-actin. G is for globular. So these tiny globes, the tiny globular structures, come together, polymerize, and form this long, thin filament, F-actin. On this F-actin, we have this tropomyosin. Tropomyosin is a protein, very, very important, because it actually covers and closes the binding sites of actin. Beneath the tropomyosin, we actually find the actin binding site. Again, the actin binding site is the part of the myosin head, and this part of this myosin head will attach and grab the filamentous actin, the F-actin, and this is covered by the tropomyosin. It actually makes sense, and we'll see why it's actually covered. On top of this tropomyosin, we have this troponin unit. Troponin is a three-component molecule, three-component protein that actually is made out of TNT. TNT is from the T, comes from the tropomyosin. Actually, it means that this the troponin is the troponin part. The TNT is attached on the tropomyosin. Then we have the TNC, the calcium binding site. This is part of the troponin that actually binds calcium. And then we have the last one, which is the TNI, the interaction that inhibits the interaction between actin and myosin. Let's talk a bit about actually what happens. Let's sum up. Again, we have the myosin that eventually will bind on the actin, and this binding site is covered by the tropomyosin. Let's keep it at that. There is this regulatory unit on the tropomyosin that controls when and how this actin binding site will open or close. Of course, the switch, let's call it, the switch of contraction is the tropomyosin, and the outlet of the interaction is the tropomyosin, and the switch is the troponin unit. So let's actually see, let's put all of these parts together to see how the contraction takes place. So we know that contraction in skeletal muscle is of voluntary movement. So of course there has to be a nerve input, a nerve impulse to actually activate this contraction. So the first point, of course, is going to be the nerve impulse. The nerve impulse is going to activate in the neuromuscular junction. We're going to have the activation in the exocytosis of acetylcholine. This is the molecule that actually activates and induces the contraction of the muscle. This acetylcholine is going to be inserted inside the sarcoplasmic reticulum and activate these tubules. These tubules are called T-tubules. What are these T-tubules? Because we need the contraction to take place throughout the whole muscle fiber in the same time, we need to be able to practically allow calcium to do its job in the whole unit, throughout the whole length of the unit of the muscle fiber. So for this reason, we have these T-tubules, these transverse tubules. The transverse tubules receive the nerve impulse and practically propagate it throughout the whole length of the muscle fiber and activate immediately the release of calcium from the sarcoplasmic reticulum. We discussed already in the cytology part what is the sarcoplasmic reticulum, but again, in summation, the sarcoplasmic reticulum is the smooth endoplasmic reticulum, and the function of the smooth endoplasmic reticulum in the muscles is to contain and to control and to store calcium. The one ion that is responsible for contraction, presence or absence of contraction, is calcium along with ATP, but the ion is fully and only calcium. So again, the nerve impulse comes from the nerve. This impulse propagates through the T-tubules, and the T-tubules activate the sarcoplasmic reticulum and allow for this calcium to go out. So what? Calcium is out. How actually does calcium allow this contraction to take place? Well, we discussed before that in the troponin unit there are 3 different units. There is the calcium binding site. Imagine it as 3 fingers like this, all right? This is going to be, let's say, for example, this is going to be the TNC, the troponin calcium binding site. Once calcium is attached on this location, it practically tilts. Imagine, of course, this is not true, but this is actually helps you imagine how it actually works. Calcium binds in the TNT, and this leads to a conformational change, which is going to be this rotation. Imagine that allows the Tropomyosin, of course, protein to practically move away from the binding site. And because of this activation, because of this movement, this conformational change, this opening, the actin filament, now the binding site of actin filament is now open, and because of this open, the myosin heads, the thick filaments, are going to be attached to this to the F-actin. Now this attachment, so we have this attachment. This is how we actually allow the attachment from the tropomyosin, from the actin and myosin. So again, calcium practically will attach on the TNC, the calcium binding site, produce and induce this conformational change. The tropomyosin is going to be placed on the side and allow for the actin binding site to be free. Once this binding site is free, then the myosin head is going to attach on this on the actin filament. So we have this attachment. So what? It attaches. The function, of course, of this the end result of this attachment is going to be the let's say the pulling actin to one specific location, towards an inward motion. Of course, this inward motion is controlled by the ATP. We'll see how and why. So once it's already bound, it will actually do this movement, this inwards movement towards the center, towards the M line. So again, ATP is the function of ATP in this case is gonna be what it allows for the myosin head to release this to stop this binding function. So after this binding and this contraction movement takes place, this inward location takes place, this is what actually will lead to contraction. How? Well, this is because, we'll talk about this in the end. This is because we have, of course, in this whole line of the thick filament and the actin filament on this top and on this top, and this towards the center. Because we have the simultaneous contraction on both ends, on both left and right, both left and right parts of the sarcomere will be contracted, will pull inwards. This pulling motion is gonna practically bring the two sides closer together. And this pulling motion towards the center makes the sarcomere shorter. This, imagine that many sarcomeres to follow in line together. The whole muscle fiber is structured with sarcomeres, and all of the sarcomeres of the whole muscle fiber will be contracted towards a central line. This contractment towards a central location is what will actually result in the contraction of the muscle. Of course, this is why when you contract, when you use a muscle, the length of this muscle, the length of this muscle actually shortens. This is because we have this inward movement of all of the filaments towards the center. This is actually a very nice picture that we can actually realize what happens at the same time. In the relaxed skeletal muscle, we have it as it is was the beginning. There is no attachment between the myosin and the actin. The thin and thick filaments are not attached and they're free. Once there's again the nerve impulse, we're going to have the calcium starting to be practically allowed and stored and freed inside the sarcoplasmic reticulum, inside the cytoplasm of the skeletal muscle. And because of this free calcium, the calcium is going to bind on the TNC portion of the troponin, lead to a conformational change, allow the tropomyosin to move away from the actin binding site, and because there is a free actin binding site, the myosin head will attach on this actin. And again, after the usage, of course, using ATP, this movement, this frees and allows the myosin heads to practically move and inwards towards the center and end up and result in this picture as it is here, which is the overlapping bands. The overlapping of the thin and the thick filaments is now complete. So the sarcomere itself shortens, means that the I band, the thin filaments will slide in and stay in this location for some seconds while the contraction lasts, and this is what causes the sarcomere to become contracted. And of course, as a consequence, these many sarcomeres together will contract and shorten the length of the muscle, allowing for contraction to take place. It's important to remember the sarcomere, the length of the sarcomere can be shortened. The sarcomere itself can be shortened, but the components are not. The length is stable. The length of the H line, the length of the I band is the same. Meaning what? Of course, this makes sense because there is no change of size in the thick filament or the thin filament. There is simply this overlapping. So the structure of the sarcomere can be shortened, but the components are stable nonetheless. Question is, how do we go back to the initial state? How do we go back to the resting state? Well, this is where ATP actually comes into place. The function of ATP is to release the myosin head from the actin site. This actually makes a lot of sense. Actually, this is why we can see this process of rigor mortis. Rigor mortis is when you have a dead person, all of the muscles are contracted in the beginning. So the body is stiff after death. And this is because there is no more ATP production. And because there is no ATP, there is no potential for these freeing of the myosin heads on the actin, from the actin. So it stays contracted. This is one. And how does it go back? Of course, the first is ATP, and the second is titin. We discussed titin right here in the beginning. 1 second. This is titin. The function of titin is, it's practically a spring-like protein. So this picture is actually very useful because titin is in a coiled, let's say, architecture. And this is because the function of titin is to function like a spring and allow for the Z disc to pull away from the thick filament. So